This is the backup plan for Thursday, March 15th. Today we are going to look at Chapter 19. So Chapter 19 has to do with viruses. So we're going to do a quick look through viruses and talk about the key components of viruses, since we are currently talking about the immune system, and the viruses traditionally are going to be kind of infecting our body, and our immune system would have to act to fight off those infections. So there's always been a lot of talk about whether viruses are actually alive or dead. So let's just first think about, well, what is a virus? Well, a virus is just an infectious particle, and it only consists of a nucleic acid. So it either has DNA or RNA. And then that nucleic acid is enclosed within inside of some type of protein coat. So there's proteins, and then there's also nucleic acids in it. That's why this was, if you think back to the Hershey and Chase experiment, that's why they used the bacteriophages, because they were able to code either the nucleic acid or the protein. And then also in some cases there tends to be a membranous envelope. That envelope traditionally comes from the host, and it's a way for the virus to kind of hide itself from the host immune system. So as I said a second ago, the viral genome is some type of nucleic acid. So it's either going to be double or single-stranded DNA, or it'll be double or single-stranded RNA. So there's 4 different options you can have here. And depending on if it has DNA as its nucleic acid or RNA as its nucleic acid is the type of virus we're going to call it. If it's a DNA virus, it of course has DNA. And if it's an RNA virus, it of course has RNA. And then the capsid is just that protein shell that will enclose that viral genome or the nucleic acid of this virus. So here you can see a couple different examples of some of the other viruses. So here we see the tobacco mosaic virus. It's kind of more of a rod shape. Here you see the adenovirus, which is more of kind of a hexagonal type shape. Here you see the membranous envelope that will surround the virus. And then here you see a bacteriophage, which this should be pretty familiar to, because you saw the bacteriophage when we looked at the Hershey and Chase experiment. So as I said before, some viruses tend to have this viral envelope that will surround the capsid. An example of one of these viruses would be the influenza virus. And traditionally these tend to be animal viruses that have this membrane coat. And so that envelope, that membrane, comes from the host membrane. So whenever the virus is released, it doesn't go through a lysis, it doesn't burst out of the cell, it actually gets slowly secreted out, kind of like exocytosis in a way, which allows it to be surrounded by membrane. And so this membrane traditionally has both viral and host molecules. Of course, it has the host molecule because it comes from the host membrane, but then it also has to have viral molecules so that it can then bind to the appropriate cell to find a new host. Bacteriophages that you guys have seen before, they're also known as just phages. These are viruses that infect bacteria, if you couldn't catch that from bacteriophages. And so what these have is they have an elongated capsid head, so this is big head that has their DNA, and then they have this protein tail that comes out from it. So what we were looking at before was a T-even virus. And so there are T-evens or T-odds. The T-evens kind of look like the Jimmy Neutron spaceship, and the T-odds kind of have more of a rod shape in the back. And so the way that these work is that they will attach to the membrane, and then they will actually inject their DNA inside, similar to what you guys saw when we looked at the Hershey and Chase experiment. So thinking about the fact that some people think that viruses are alive and some think that they're not. Viruses are an obligate intracellular parasite. So obligate means that it is unable to survive without a host. It's kind of obligated to be with that host. And then intracellular means that it's a parasite within the cell. And so in order for the virus to be able to reproduce, and in order for the virus to be able to do anything, it needs a host. So if there's not a host, the virus is just free floating, waiting to find a host. It doesn't metabolize. It isn't able to reproduce on its own. it doesn't grow, it doesn't divide, it doesn't do any of that unless it has a host. Um, and so each virus has a certain kind of host range. There's only certain hosts that the virus can actually infect. Um, and so kind of it also has to do with the cells of the organism. So there was this big thing with the bird flu and the swine flu. Um, and it's really cool because the reason why we're able to get swine flu is because pigs and chimpanzees, they share a similar cell within their epithelial tissue of their trachea. And then we share a type of cell with the chimpanzee. And so when the swine flu went into the chimp, it mutated and was able to then adhere to a new type of cell, which is why we're able to now get swine flu. So it's kind of cool. So as I said before, the host cell is going to provide all of the resources for the virus to be able to replicate. So once that viral genome has entered the cell isn't really able to recognize that this is viral nucleic acid versus the host nucleic acid. So what it does is it's going to use that DNA, make RNA, and of course make proteins, or if it's already in an RNA form, it will then also make the proteins. So the virus makes use of any of the enzymes within the host. So the DNA polymerase, the RNA polymerase, the helicase, the topoisomerase, all of these enzymes that are important for replication and transcription and translation. It uses all those. It uses the ribosomes, it uses the tRNA, it uses the host amino acids, it uses the ATP. Everything that the host has is what is being used by the virus. And so what's really cool though is once you make the protein is able to self-assemble with the nucleic acid just to spontaneously make the new viruses. So like if you were to have a test tube and you were to put the nucleic acids in there and you were to put in the viral proteins, they will automatically bind together and form new viruses, which is really cool. So here you can see the kind of simplified version of the viral reproductive cycle. So the first step would be for the virus to enter the cell and then uncoat. So of course the capsid and the DNA have to separate from each other. That viral DNA can do one of two things. It can either get replicated, so here you see the replicated DNA, which will then also be used later to make our future viruses, or that DNA can get transcribed. So here you see the mRNA, and then it can get translated. Here you see our capsid proteins. And then once you have those capsid proteins and the DNA, it's going to self-assemble to make new virus particles that will then exit the cell and kind of come out. So there's two different cycles that a virus can do. The lytic cycle is actually going to cause the death of the host cell. So thinking about this, the virus isn't going to want to overly infect the host cells because that's going to cause an over death of the host. Because if every single time that you replicate, you kill your host, you're sooner or later not going to have any hosts. And so there is another cycle that the cell will go through. But for right now, we're going to look at the lytic cycle. And so if you've ever read Twilight, like they talk about how the Renesmee, she kind of ate her way out, or she was going to have to eat her way out of Bella. Well, the same idea is happening here. These new phages, they release an enzyme, and that enzyme basically digests the cell wall of the host, which causes a hole in the wall. And so once that hole in the wall happens, those virus particles can come out and all this water rushes in, which of course causes the lysis of the cell. And so the cell can't survive past that. And so any phage that's able to reproduce by the lytic cycle is actually called the virulent phage. And bacteria have ways that they can defend against phages. We talked about these back in Unit 4B. We talked about those restriction enzymes, and that the restriction enzymes would recognize viral DNA, and it would recognize, and it would of course cut up that viral DNA to inhibit this process. But this is how the bacteriophages reproduce. So here we see a diagram. Here you can see our T even right here. It's going to attach to the bacteria. It's then going to inject the nucleic acid, which in this case is DNA, into the cell. The cell is then going to use its RNA polymerase and its DNA polymerase and the ribosomes in order to make the viral genome and make the protein. It's going to self-assemble, and then it's going to release this enzyme that causes a hole in the wall, which causes the cell to lyse and burst open. And then all of these cells are then going to find a new bacteria to attach to, and the process occurs again. So as I said before, the virus doesn't want to kill off all the hosts. It needs to find a way that it can coexist with the host and allow the host to continue to make copies of it without damaging or hurting the host. Because once you run out of hosts, of course, the virus can't replicate anymore. And so phages can either go through that lytic cycle, or they can do another cycle called the lysogenic cycle, which makes it a temperate phage. And so what will happen is that the virus will insert itself into the host genome. So it actually will make its virus genome part of the host genome. And I'll show you a picture in a second. Unless there's some type of environmental signal which causes it to exit out of the host genome, and then it switches back into this lytic phase. And so you kind of see that the cells go back and forth between these two processes. So here you can kind of see that temperate phase, in which here we have a T-odd, it kind of looks like a rod. It's going to attach, it's going to inject its viral DNA into the host cell, right? So there's one of two things that can happen. That DNA can either go through the lytic cycle, in which we're going to, make the new DNA. It's going to lyse the cell, which makes the new phages, it then infects the new cell. Or another process is it can actually cut this host DNA and it can insert itself into that host. And so every time that that bacteria reproduces and makes another copy of the bacteria, that virus is part of that host genome. And so it's going to continuously make copies of the viral DNA until some type of environmental stimulus happened. So if you remember back to when we talked about the herpes virus and HIV, these two viruses all do this exact same thing. They insert themselves into that host genome. And so every time you make copies, let's say the HIV virus, every time you make a new T cell, then you're making copies of that HIV virus that has embedded itself into that T cell's genome. And so that's why HIV takes so long to really affect you, is because you only make multiple copies of it when you're sick. So when we think about classifying animal viruses, there's two ways we classify it. We classify whether the genome is single-stranded or double-stranded, as we mentioned before. We also classify whether it's going to be DNA or RNA. So we have a couple examples just to show you how these different viruses are classified, and also give you a couple examples of the different classifications. So here you can see a couple examples of double-stranded DNA, single-stranded DNA, and double-stranded RNA. So double-stranded RNA, an example would be, of course, our herpes virus, which is herpes simplex 1 and 2, which would be our cold sores, our genital sores, all that fun stuff. Epstein-Barr, which is your mononucleolus. Pox virus, which would be smallpox and cowpox. Examples of a single-stranded DNA would be the parvovirus, which causes of course, there's a mild rash. And then an example of a double-stranded RNA would be the Colorado tick fever virus. Lots of fun there. And I'll show you some more on the next slide. Some examples of single-stranded RNA you can see here, of course, single-stranded RNA that use it as a template for RNA synthesis, and then also single-stranded that use it for DNA synthesis. So single-stranded RNA, you can see here we have hepatitis C, the yellow fever virus, rubella. You guys all got a shot for that when you were a kid, a vaccine for that. You also see that we have the rabies virus is a single-stranded RNA, as well as Ebola is. And then last but not least, you see that HIV would be a retrovirus. It has a single-stranded RNA, and it actually uses that as a template to make DNA. So many of the viruses that we have that affect animals, they tend to have a membranous envelope, as I mentioned before. And so this membranous envelope traditionally comes from the host cell. And so what ends up happening is during kind of an exocytosis type process, the virus will bud off from the host cell, allowing it to have an envelope of the host cell. And then it also will have glycoproteins from the virus, which will allow it to be able to bind to the specific receptors on its next host cell. And so the same way that our membrane proteins are made, the viral protein membranes are made. So the viral proteins will be synthesized in the rough ER, and then they're going to go to the Golgi, just like we've seen before. So here you can see a kind of a visual of what I was talking about. So here would be our virus. And so when it goes into the cell, it has the capsid. It also has the viral DNA inside. The viral genome is, let's pretend it's an RNA. That's going to act as a template to make mRNA, so we can then make those glycoproteins in the ER. And then we also see that we can make copies of that of that RNA for the new genome. And so what ends up happening is now we have these viral proteins on the outside. We also have our new virus here budding off. And so now we see we have our new assembled virus. So back in Unit 4B, I believe, we talked about these things called retroviruses. And these viruses were ones that went against the central dogma. Central dogma traditionally says you have DNA, which helps you produce RNA, which then produces the polypeptide. Well, retroviruses have an RNA genome. And so what they do is they use this enzyme called reverse transcriptase, and that allows their RNA to make a pair, and then they make it into kind of a DNA. And then that viral DNA gets incorporated into the host genome. So it becomes this thing called a provirus. And it's now a permanent resident in that host cell. So every time that the host replicates and makes a copy of its DNA, it makes a copy of that virus as well. And then the host RNA polymerase, of course, transcribes that DNA into RNA. And then that RNA will then, of course, make more viruses. So here you can see the example of that. So here we have our host cell, and here we have our virus that has come in. Okay. So here you see that the nucleic information is RNA. So the RNA is going to go and make a RNA DNA hybrid. So by base pairing, we make a DNA copy of it, and then we're able to make a double-stranded DNA. It then moves into the nucleus, it cuts into the host genome, and it adds itself in. And then once you go through transcription, you're going to transcribe copies of the new virus. And so then that virus can then be released out. So as we said before, viruses traditionally can damage or kill their cells because of the fact that they can cause the release of different hydrolytic enzymes from the lysosomes. Other things that viruses do is that they can actually cause the release of different toxins. So, for example, in scarlet fever, it causes a release of toxins, which causes you to get a rash on your chest from the scarlet fever. For other ones, the envelope proteins themselves are toxic, and so they harm the cells just by the envelope proteins. So as we talked about before, vaccines are traditionally harmless derivatives of that pathogen. So it's either pieces of the virus, it's dead virus or like non-functioning virus, or it's weakened virus. And so what that does is it gives the immune system kind of a stimulation so that it can start fighting the virus, so that if you were to come in contact with that virus again, you would know how to fight it and then you wouldn't be infected by it. And so vaccines are used to prevent a lot of different viral illnesses, like you guys got one for chickenpox when you were a kid. You probably got hepatitis B shots that stopped you from getting hepatitis B. You probably got the MMR, the measles, mumps, and rubella. So you've gotten a lot of vaccines throughout your lifetime. But the big thing to note is that viruses cannot be treated by antibiotics. So if you've ever had mono, they've always told you just got to let it run its course. if they give you an antibiotic when you have a virus, what they're doing is they're trying to kill the bacteria that the virus is infecting. But they do make these things called antiviral drugs. And what they do is that they will kind of treat the virus, so it kind of inhibits the virus from working, but it's not actually a cure. So for example, I had chickenpox when I was a kid, and although I don't currently have chickenpox still lives in the base of my spine. And so when I get older, there's a chance that I could get shingles. And so shingles is kind of like an adult form of chickenpox in a way, and it usually affects elderly. So you never really get rid of your virus. You kind of are just treating it to stop it from kind of reproducing at the time. So that brings us to the end of chapter 19. So you guys should right now go and work on your chapter 19 reading guide.